Guys, until now, we have explored the metabolism of rather simpler carbohydrates. Simple in the sense that they are made up of, usually they are made, what we have discussed until now are those carbohydrates which are either made up of simple sugars like glucose or fructose, or even if they are made up of many monomers, for instance like glycogen, which is a polysaccharide, still at the end of the day they are carbohydrate moieties. Now today let's we discuss little bit about the metabolism of those carbohydrates which are conjugated with other moieties also, like the carbohydrates which are conjugated with proteins known as, for instance, glycoproteins or peptidoglycans. As far as peptidoglycans are concerned, although they are not as frequently used in the energy dynamics, I mean they are not source of energy in the sense or in the same way as glucose or lipids are, but still they are very important structural as far as functional molecules. Peptidoglycans are, of course, as the name shows, that's made up of both the carbohydrate moiety and the protein moiety. They are very large molecules. The carbohydrate moiety is known as GAGs or glucoaminoglycans, and we will study in detail about their metabolism and what role they are playing. Keep in mind that students usually feel little bit of, I mean this topic for them is a little bit difficult because it involves many complex molecules. So I want your real focus on this topic. So as I have just mentioned that whenever we are talking about proteoglycans, that is the carbohydrates which has protein moieties, the major or the most important carbohydrate moiety is GAGs or glucoaminoglycans. The composition of glucoaminoglycans is not very complicated, but it's quite different as compared to the carbohydrates you have studied until now, that is the glycogen as a polysaccharide or the oligosaccharides or disaccharides in which the glucose is the main monomer. So as far as composition of GAGs is concerned, they are unbranched heteropolysaccharide chains. What is meant by heteropolysaccharide? That in contrast to glycogen, which has only one sort of or one type of monosaccharides, that's glucose, here the units, the repeating units are made up of two different type of hexose sugars. These two repeating units are, one of them is the amino sugar and the other is the acidic sugar. The amino sugars are D-glucosamine and D-galactosamine. These both sugars have the tendency to get first acetylated, giving rise to the formation of N-acetyl glucosamine and N-acetyl galactosamine, and then they are also sulfonated. That is, addition of sulfur also takes place in these moieties. Acidic sugars, on the other hand, they are composed of D-glucuronic acid and L-hyaluronic acid. They have negative charges and they give, as a result of this, overall negative charge to the glycoproteins or to the proteoglycans. So because of this negative charge on the GAGs, what happened that the overall charge on the whole molecule is negative, which have its own functional importance, not only structural importance. The GAGs have a unique water binding ability because they are quite polar, as I have already told you that they have overall negative charge. They are very important component of the ECM, that is extracellular matrix, the space between cells. So they occupy that space. They are playing very important role in the extracellular matrix. They are adhesive proteins there. They, for example, fibronectin are very important there, which are playing very important role in connecting the tissues. Within ECM, they also give a structural support, of course, to the ECM because of their very, what we say, their flexible nature. They are also important for interaction with other molecules. They, because of this flexible nature, you can find them making a very important component of molecules or substances which are very fluid in nature, like mucus, which is very lubricating. The negative charge on these molecules, it has a specific, it gives a specific feature to these molecules, that because of the negative charge, these molecules repel each other. And because of this repulsion, they tend to, when you keep them close to each other, they tend to slide over each other. It's like a magnet. When you bring a magnet close to each other, the same pole of the magnet, what will happen that as a result, in the same way as the magnet, or as a result of the repulsion between the same poles of the magnet will look like sliding over each other. Similarly, they'll slide over each other, which makes them very lubricating, very slimy. So this is, you see in your mucus, that's why it's so slimy. But it has its very functional, importances also. Similarly, your tears also contain these compounds because, again, lubrication is needed for your eyes. The classification of these compounds depend upon the type of sugar moieties which are involved in them, the sulfate units, how much sulfate units are added to, and to which compound the sulfate units are added, and the overall distribution. They have an exceptional compound or type known as hyaluronic acid. It's exceptional in the sense that it doesn't have sulfonation with it. Sulfate moiety is not added on them.